2 years ago, Sir Alan McClay introduced the first McClay Foundation lecture. He laid out his vision for translational research that not only aimed at publication, but also made a real difference to patient care. Sadly, he's no longer with us, but his passion for research and innovation continues through the McClay Foundation, a charity whose aims are to advance the use of diagnostic tools and drugs in the prevention, control and cure of disease, to support and encourage research and innovation, to ensure the latest advances in healthcare are available to all, including developing countries, and to generate and promote employment opportunities for all. Consistent with these aims, our next speaker has made a significant contribution to patient care over the last 2 decades. Dr. Goldberg was educated at Harvard, Emory and Georgetown universities. Throughout his career, he's had several key roles in the American Society of Clinical Oncology, North Central Cancer Treatment Group, and the Cancer and Acute Leukemia Group B. Currently, he is the Professor of Gastrointestinal Oncology, Division Chief of Hematology and Oncology, Physician-in-Chief of the North Carolina Cancer Hospital, and Associate Director for Clinical Research at the Lineberg Comprehensive Cancer Center. He is the Associate Group Chair of the Gastrointestinal Cancer Committee for CALGB and is on the editorial boards of a number of peer-reviewed journals. He has published more than 400 scientific articles, many of which have been highly influential in the management of gastrointestinal cancers. He remains actively involved in clinical trials testing new chemotherapy and the application of pharmacogenetics to personalised patient care. Dr. Goldberg, you're very welcome to Belfast, and we look forward to your talk.
Thank you for the kind introduction and for the privilege of this podium. And what I'm going to talk to you about today is maximizing the harvest. We've been talking a lot about clinical trials all morning long for the last several days. I had the good fortune to run a positive clinical trial. We have used that clinical trial to learn many things, and I'm going to illustrate some of them today. Even negative clinical trials can be useful if we don't just put our heads down in disappointment and walk away from them. And I hope to illustrate that as well. Now, what is a parable? A parable, according to Wikipedia, is a succinct story in prose or verse. I tried to put mine in iambic pentameter, but gave up, which illustrates one or more instructive principles or lessons, or sometimes a normative principle. It sketches a setting, describes an action, and shows the results. Now, this brings me back to the individual whose name we are recognizing as the sponsor of this meeting, Sir Alan McClay, in the setting of Northern Ireland, which at the time that he was an active businessman was not considered necessarily to be a hospitable environment for growing business. He founded Galen and then Almac. He has been described as the most significant philanthropist Northern Ireland has ever known. And if you walk around the campus at Queen's, you'll find his name, which I'm told he was reluctant to have inscribed on a number of buildings, including the McClay Library. Some descriptors that I picked up from a BBC special that Patty sent me a disc of were that he was homegrown, a child of Northern Ireland, that he had humble roots growing up on a farm, that what he did was transforming in Northern Ireland because he kept his core values, built on those values, probably had some luck along the way, but ended up being a significant force in the lives of many. And one of the things that I really enjoyed about the special, which you I'm sure can find on the BBC website, was the description of his long-time relationship with Lady McClay, which he described in a setting filmed in his living room as a perpetual courtship. And some of you may know the story that they were companions for many years before they became husband and wife. And in many ways, our relationships with each other, I think, can be described as perpetual courtships with our friends, with our scientific colleagues. Part of the joy of being a human being is the ability to interact with each other and to share that interaction in a positive way that creates new knowledge and energy. And I think that that is why Sir Alan's legacy has been outstanding, organic, and growing. And for those of you who didn't have the opportunity to meet him, this is what he looks like. So in the beginning, there was 5FU. And it was administered according to one of two programs, the Mayo Clinic program, which was 5 days in a row of 5FU. You put the drug in, and then you had to deal with the toxicity later, versus the Roswell Park regimen, where you gave the treatment weekly, and at least you could adjust as you went along the way, versus the 5FU regimens that had become popular in Europe, which were infusional regimens. And there was a virtual wall between infusional regimens and bolus regimens that went somewhere through the middle of the Atlantic. We never used infusion, and people in Europe seldom used bolus. In early 2000, we finally got a second drug for colorectal cancer. That was irinotecan. And in the United States, at the time that this trial, 9741, was conceived in 1997, the standard of care was transitioning from 5FU and leucovorin to a bolus-based IFL regimen, irinotecan 5FU leucovorin. And when the study first began, this was what it looked like. And you'll remember, if you're familiar with 9741, that it is the study that led to the approval of oxaliplatin in the United States for the indication of first-line treatment of metastatic colorectal cancer. When it first began, there was no oxaliplatin in the study. It really was a drug scheduling study looking at the 5FU leucovorin control regimen, irinotecan based on a bolus 5-day-in-a-row Mayo-based regimen, the SALTS regimen, which was based around the Roswell Park program, or we were going to dabble in infusion in the United States using a 24-hour infusion regimen favored by our German colleagues in the AIO. And many were skeptical of this, thinking that Americans could never transition to infusion. But the study went forward and was supported by the entire might of the intergroup collaboration. So CLGB, ECOG, NCIC, and SWOG joined it. At its peak, it enrolled about 130 patients a month. And that's relevant because these were enrolled in about 105 centers across the U.S. and Canada. And it was very difficult to know what exactly was going on with those patients in real time. And that proved to be an issue for us, as I'll talk about. This study has been good for my CV and the CV of many other people. There have been literally hundreds of individuals who have authored and co-authored papers that have come from this, a total of 32 papers to date, one of which was just published in The Oncologist in the last month, and more than that in terms of abstracts. And according to Google Scholar, if you look at the primary paper published in JCO in 2004, it's now been cited in almost 1,300 articles. So this study has had an impact, at least in filling journals, if nothing else. The topics that I'd like to talk about today include some that relate to clinical trial and study design, hoping to talk not just about this particular study, but how this study relates to the art and science of trial design. How it, in 1997, became one of the earlier studies to focus on translational science. How we helped to develop the science of meta-analysis and pooled analysis in colorectal cancer by not just offering our data up to others, but collaborating with others who had large data sets in order to gain insights that couldn't be gained from single trials. And also that we looked at what we were doing as we were doing it and related that to clinical trials methodology. Now, one of the important things that we did in this study was novel at the time, although standard currently, and that is to include baseline quality of life data. And this was done at intervals, but started out before the patient actually even got their first drug at the time they enrolled. And we chose validated mechanisms for doing this. It took about 20 minutes for the patient to do at selected visits, looking at the symptom distress scale and also a quick and dirty approach to estimating quality of life called the Uniscale. And perhaps this would be a good thing for each of us to fill out at the end of every day as we get ready for tomorrow. Now, as this study began, there was information coming from Francois Levy and from Aymeri Degermont in France that oxaliplatin was a highly active drug in colorectal cancer when given with 5-FU. And so Dan Sargent and I met with the leaders at the National Cancer Institute trying to determine if there was a way that we could quickly import oxaliplatin into America and get some experience with this drug. And what we decided, much to the dismay of Pharmacia, who owned irinotecan at the time, and was supporting the trial by giving irinotecan, was to modify 9741. We've talked a lot this morning about adaptive trial design. In fact, I was a bit amused and surprised by how often that was talked about this morning. This is a picture of Dan Sargent, who is the lead statistician on this study. His mentor is Don Berry from MD Anderson, who pioneered the adaptive trial design. And just to show you how important accidents are in life, Dan was an undergraduate at the University of Minnesota. He took a course from Don Berry when he was a professor there. At the end of his senior year, Dan wasn't sure what he was going to do. He was walking across campus and ran into Don Berry, and Don said, "What do you mean you're not sure what you're going to do? You're going to be a clinical trial statistician." And on the spot, he offered Dan a spot in his graduate program, which is what we call Bayesian recruitment. We didn't design this trial to be adaptive. In fact, the notion of adaptive trial design really didn't exist in 1997. The definition of it, again, taken from Wikipedia, is a study that allows modification of ongoing trials while aiming to preserve the statistical validity and integrity of the study. Before I turn from this slide, I also want you to notice one other thing about Dan. Those of you who are clinicians in here might notice that he has a very small collar size. And that's relevant to what I'm going to talk about in a moment, because he's a survivor of childhood Hodgkin's disease and got mantle radiation, which means that he has a sparse beard and a small neck. But he'd been on the wrong side of the chemotherapy infusion pump as a teenager. So in our adaptive design, which some described as the Mr. Potato Head approach to clinical trials, and you may not know what Mr. Potato Head is here in Ireland, but in America we play with our food sometimes. And Mr. Potato Head was a game that I played with as a child where you could poke eyes and ears and nose and arms into a potato and then take them off and modify it. So the reason people called this the Mr. Potato Head study was that we kept putting arms on and taking arms off as the study was going on, and now we call that adaptive trial design. So this was the 6-arm version of the study, where we brought in IFL as the standard of care. At that time, we still had 5-FU leucovorin in there because IFL had not been accepted as the new control arm. And then we had FOLFOX, a non-5-FU containing regimen, which was historic at the time in colorectal cancer. And then, because I was at Mayo, we had several different variations on the theme using the Mayo bolus approach to 5-FU. Well, Dan and I would meet every week to talk about how the study was going. And Dan got nervous about it because he felt that he really didn't have a sense of how things were going in those 110 odd centers where patients were being treated. And we would get occasional reports of toxicity. They often would come in 2 months after the event occurred. And there was no real way to have a sense in real time of what was going on out there. So we developed a simple single-page report that we required centers to fax into us within 24 hours of a grade 4 toxicity, a grade 5 toxicity, meaning a patient death related to treatment, or a hospitalization. And each night this was entered into the database, and each morning I would get an email, and Dan would get an email with any new events, plus all the cumulative events that had occurred on that arm. And the advantage to that is that we really were able to watch what was happening with a 24-hour lag time. And we were able to have some cumulative data about it. And this is very different than the way we ran clinical trials over the years. They were to some degree a black box. You had a flurry of activity where you put together the study and got it approved and through all the regulatory hurdles. Then you opened it and waited for all the data to come in and then analyzed it. Well, that wasn't the way we did this. And it's fortunate that that wasn't the way we did this, because one of the things that surprised us was that we found that when you started to add other cytotoxics to bolus-based 5-FU, there was an excess of early patient deaths. And here you can see some data from that. Now, the notion of a 60-day mortality rate is a notion that Dan borrowed from his former work in AIDS clinical trials. It gives you a pretty quick reading on what's going on. In colorectal cancer, almost all patients are performance score 0 or 1, even in advanced disease trials. They seldom die of progressive disease within 60 days. So this sort of metric would be a measure of toxicity, not of colon cancer mortality. And it was very surprising to us to see this very high toxicity rate. But what was even more surprising was to see this in the then standard of care, IFL. And to remind you, at the time, IFL had only been tested in one randomized trial, a trial run by Pharmacia. There was no real good data about severe toxicity, even though this regimen was being adopted rapidly countrywide. We also then looked at the toxicity on the 2 oxaliplatin arms and found that that was lower, the 60-day mortality. This has now become a metric that is widely reported in colon cancer trials. So as a consequence of that, we dropped the bolus arms with the Mayo schedule, and the Mr. Potato Head study for a while had 3 arms. And these were the 3 arms that are the main subject of the study. And the outcome of this ended up being reported early because this study crossed early stopping boundaries specified in the protocol as administered by the Data Safety Monitoring Committee. And what you can see here was that IFL, which is the salts regimen, had a 31% response rate. That was about what it was in the trial that Lenny reported in the New England Journal, while Folfox had a 45% response rate, which was similar to what Amory was reporting in his studies in France and Europe. And IROX had an intermediate response rate. If one looks at the progression-free survival endpoint, clearly Folfox outdistanced the other 2 arms of the study. And if one looks at overall survival, we had moved survival from a median of about 12 months with the 5-FU leucovorin regimens to about 15 months with the IFL regimen, and this moved it up to about 20 months, which may seem like a small difference, But again, I'll remind you that median survivals are made up of people who often have very long survivals, as well as people who have meet the median. And the other thing that was important about this was that toxicity, with the exception of neutropenia and neuropathy, favored FOLFOX in all accounts. And people don't die of neuropathy. They occasionally die of neutropenia, but fortunately, the neutropenia did not translate into neutropenic fever very commonly, as you can see over here. So the initial results of this study, which were presented at ASCO, led the FDA to approve FOLFOX for first-line colorectal cancer therapy in the United States. It changed the standard of care in the United States, more so than in Europe, where irinotecan is still commonly used in combination with 5-FU as a first-line regimen. And it led to robust sales of oxaliplatin around the world. It also led us to the notion that giving all cytotoxics to patients was optimal who had metastatic colorectal cancer, and that we ought to relegate bolus-based 5-FU regimens to historical interest. And this led to wholesale adoption in the United States, which they said couldn't be done, of infusional 5-FU. And this is data from a paper that Axel Grothe, Dan, and I published in JCO, showing that on the left-hand side, median overall survival, on the bottom, percentage of patients with all three drugs, and basically the more patients got all three drugs in the course of management of metastatic colon cancer, the longer they lived. Here's FOLFOXIRI, where you guarantee that patients get all three drugs because they get them up front, and it's off the curve. Now, at the time that this study was reported, there were some criticisms of it. One was that the comparator arm was bolus 5-FU in the irinotecan arm, but of course that was the standard of care in the US, and so that's what the FDA mandated. The other was that there was not equal access to oxaliplatin for second-line therapy, as oxaliplatin was highly restricted in the United States at the time of this treatment. And so some thought that the absence of access to oxaliplatin might have affected the outcome. And so this additional modification of the trial helped to deal with that. What we did was we modified IFL because we were having so much toxicity and finding that people were reducing doses of the irinotecan according to protocol within the first 6 weeks. So we just dropped the doses, dropping the irinotecan from 125 to 100. And compared that to FOLFOX in hopes that with less toxicity, we would find irinotecan worked better. We found exactly what we had found previously, that FOLFOX still was advantageous with a highly significant p-value. And here you can see the time to progression and the overall survival data. We did succeed in reducing the toxicity of the regimen, so that was a positive, and I think people who then continued to use IFL in future years were influenced to reduce the initial starting dose. But one of the accidental but very helpful outcomes of this was that in second-line treatment, if you look at the patients who got IFL, 74% of them got any second-line treatment, and 56% of those got oxaliplatin. In the patients who got FOLFOX up front, 75% got second-line treatment, and 56% of them got irinotecan. So this essentially answered the issue of whether the advantage for FOLFOX in the three-arm section of the study was due to lack of availability of oxaliplatin in the second line. So the dose intensity, as I mentioned, was actually much more practical with the reduced dose IFL, and it delivered about the same doses without the initial worry of toxicity. So the conclusions were that FOLFOX was better than reduced dose IFL as well, and that there was equal access to second line. Now, we're all commonly used to doing liver resections in our patients with metastatic colorectal cancer. But in the late 90s and early after the turn of the century, this was not very commonly done. Again, there were reports from Henri Bismuth and René Adam in Paris that using surgery for limited liver metastatic disease was valuable. But Americans were slower to take that on. And here you can see the sort of responses that we were seeing with FOLFOX, where a lesion like this would be reduced to a lesion like that. And this got American surgeons to take notice. Let's see. And surgical techniques were improving so that you could remove 3 segments of the liver or do a trisegmentectomy and still have a living patient. Now, we didn't specify that patients should have surgery in this circumstance, but we did allow it. And you can see that of the 800 patients enrolled in the 3-arm part of the trial, about 25 patients underwent surgery. Almost all of them were patients who had been treated with oxaliplatin. And for those patients who were resected, the median overall survival was much longer than had been expected, and about a third of those patients ended up being long-term survivors. And this was an early experience in the United States that has now led to a change in practice, not solely because of this trial, but because of the work of many people. Next, I'd like to turn to clinical trials methodology. And the business of doing phase 3 clinical trials is a complicated business. It's incredibly expensive. Often clinical trials that enroll 1,000 or 1,500 patients will cost a drug company in excess of $50 million. And some of that is because of regulatory requirements. And being smart about what we require, I think, is certainly opportune, particularly in today's economy. So we looked at this trial, and in the end, this trial, through its iterations, recruited almost 1,800 patients, so we had a large sample size to look at. Some of the things that we looked at, which I'll give you some details on in future slides, were, is it really useful to report grade 1 and 2 toxicities? How many lesions do you need to measure? You remember the RECIST criteria is originally constituted requiring measuring up to 10 lesions. Do we really need to do follow-up CT scans? Or could we do 1 or 2 tumor measurements and use that as our surrogate endpoint? And one of the other things that we looked at is, in the age of bevacizumab, whether targeted therapy really was so advantageous compared to cytotoxic therapy. So let me talk to you a little bit about some of these methodologic studies. This is a study where the first author is a master's level statistician at Mayo Clinic, Michelle Mahoney. It was an assessment of routine adverse event data on North Central trials. It was dominated by data from the 9741 trial. As you remember, adverse events are something we're asked to look at every time we see a patient. They're graded from 0, meaning not present, to 5, meaning the adverse event resulted in the death of a patient. And doctors were asked to relate them as possible, probable, or definitely due to drug treatment versus unrelated or unlikely. And this is the definition of toxicity. An adverse event may actually not be related to treatment. So in this particular data set, we had data from about 1,200 patients. Believe it or not, there were 75,000 adverse events reported. And often these required us to go back to the data monitors and say, well, you forgot to tell us what the grade was, or whether the doctor thought it was definitely or possibly related, or you didn't send us in the right form. And then we looked at it and said, well, how many of these adverse events were really relevant? And the vast majority of them were grade 0, 1, or 2. Only 3% of them were grade 3 or more, which is what really matters. But yet, despite that, there were 8,300 queries that had to go back and forth and back and forth to close out the charts. Each study that we looked at had specified on average 12 types of toxicity they wanted to know about. What we concluded in this was that much of what we were forced to do was make work, and it really didn't contribute to what was going on. And so actually this led to the NCI liberalizing criteria for adverse event reporting. Now, what about RESIST? Well, you'll remember that RESIST was a consensus document made up of groups from all over the world who came together and said, this is how we ought to measure tumors for response rate and progression-free survival. It was intended for tumors where response was the primary endpoint, and it was meant to standardize definitions. Initially, we used to measure lesions bidimensionally. This allowed us the great liberty of only doing unidimensional measurements, but required 10 measurements. And trying to get your radiologist to measure 10 things the same way every time led to arguments in back hallways in the radiology suite. So what we did was we looked at objective status by cycle, confirmed response, date of progression, and tried to look at evaluations of agreement between all these different endpoints versus the first and largest study. We pooled data from 33 trials, including about 2,500 patients, some of which were before and some of which were after RESIST. And what we found was that if you measured the longest 2 or the longest 3, there was tremendous agreement on progression date among patients with 5 or more lesions. If you looked at confirming the response, similarly, there was tremendous agreement just measuring 1, 2, or 3 lesions. And based on our data, we thought that measuring any more than 2 lesions was unnecessary. So this went back to the RESIST committee that then went on to formulate RESIST-2, and they heard our message. And now you only have to measure 3 lesions instead of 10 lesions. So this was a useful thing that changed clinical trial methodology, at least in the US and in places that use this. Now one could then ask the question, well, do we need to confirm responses? And so we looked at this data, and this study was just published in The Oncologist. Tumor status at 12 weeks predicts survival in advanced colorectal cancer. So we looked at measurements to compare response, stable progression versus response or progression, and we evaluated all scans, 12 and 24-week scans, to predict overall survival. And the bottom line was that what we found was, let me go back here, that the use of actual measurements was no better than a single binary or 3-category measurement at predicting overall survival. And so you could potentially measure one thing at 12 weeks as a response in colorectal cancer. Now, what about using response rate as a surrogate for clinical benefit on progression-free and overall survival in metastatic colorectal cancer? This was a collaboration we did with Genentech, where we looked at the Hurwitz trial of IFL plus or minus bevacizumab and compared that to the outcomes in 9741. So you can see that there was 3 arms in both of these studies. We only looked at 2 of the 3 arms. We had 1 arm in common on both studies. Here the experimental arm was FOLFOX. Here the experimental arm had bevacizumab added to IFL. What we found was that not just the responders benefit, but even non-responders benefited somewhat in these studies. Here you can see the PFS for non-responders, and it didn't matter whether you were getting a targeted agent or just chemotherapy. And here's the overall survival data showing the same thing. This was statistically significant in all areas. So in both studies, patients without response still benefited from the superior regimen. So stable disease is a valid endpoint and is of value to patients in terms of overall survival. The magnitude of benefit was similar for responders and non-responders, regardless of whether it was chemo alone or with a targeted agent. Progression-free survival and the percentage of patients experiencing tumor control may actually be more accurate in reflecting patient benefit than responses. Now, the other thing that we looked at was we've talked so much in this meeting about heterogeneity of patients with colorectal cancer. In the beginning, heterogeneity was something that we understood based more on amount of disease and on patient performance score and things like that, patient characteristics rather than tumor characteristics. And this was an analysis that we did looking at the data from 9741. Now, one of the things that we reported in this to make a point that I made earlier was that even though the median survival in this FOLFOX arm was about 20 months, we had about 1 in 10 patients that was alive at 5 years, some of those alive without disease. And that's a new milestone for colorectal cancer. Obviously, there are a number of patients that progress rapidly, don't benefit from chemotherapy, and die within a year of diagnosis. There may be several ways that we can predict that, one being performance score, another being some biochemical abnormalities, another being a bulk of disease in the liver or elsewhere. But at the time that we did this, really no baseline factors were really being regularly used. At the time we did this analysis, there was an important article in JAMA advocating the use of risk stratified analyses that uses pre-existing risk models to divide patients into risk groups. So to give you an example of this, if you have a study that restricts enrollment of metastatic colorectal cancer patients to PS0 patients, the outcomes are likely to be much better than if you allow PS1 or PS2 patients, and I'll show you some data on that in a minute. But that wasn't necessarily something that people reported. Now, Henning Köhne from Germany had put together a model looking at performance score, white blood count, alkaline phosphatase, a number of sites of metastatic disease, and we looked at his model in our study. What we found was that by using this risk stratified model, you could make distinctions between low, intermediate, and high-risk patients. The median survivals were very different, illustrating the point that if you only enrolled low-risk people in your trial, your survival stats would look very good. If you enrolled high-risk people, your survival stats could look very bad, and it would be worthwhile to have some sense of the makeup of the population in a trial. This is the results for overall survival showing similar outcomes. In our analysis, FOLFOX was superior to IFL in all risk groups, including the poor risk group. So this is also another potentially useful way of making sure that when we compare across trials that we know what we're comparing. Now, we did a number of pooled analyses as well using the data from this study, and I'm going to talk about just a few of them. One of them looking at older patients, another looking at PS2 patients, and then one just published again this year in Journal of Clinical Oncology looking at the very young. So this was a study published in 2006 in JCO. We're going to focus on two studies here, the first-line study by De Gramont and the 9741 study that I did. We also, in this analysis, looked at the adjuvant and second-line trials. Here's data in elderly patients for first line showing the age less than 70 patients on FOLFOX versus control. and the age greater than 70 patients on FOLFOX versus control. And the shapes of these curves are very similar. The hazard ratios very similar, indicating that the older than 70 patients had similar benefit to FOLFOX as did the younger than 70. Here is overall survival showing similar findings. In terms of toxicity, we found more hematologic toxicity, significantly more. In the older patients' marrow is less resilient than that of younger patients, but this was not clinically significant toxicity, rather it was laboratory-based. Other things like fatigue, neurotox, diarrhea, nausea and vomiting, and 60-day mortality were not significantly different among the older versus younger patients. What about performance score? Well, we all know the first day we sit with a patient in clinic who's now been diagnosed with metastatic colon cancer, who's sick, that we have anxiety about how they're going to do with combination chemotherapy. And so here's some data showing the difference between overall survival in patients who are performance score 0-1 versus those with performance score 2. And median survival of about half in the PS2 patients. So the question then becomes, well, ought we to be treating these patients aggressively? And here you can see that for patients who were treated with FOLFOX, even though they were PS2, they did better with the better treatment, even though the advantage that they gained was much less than the advantage gained by those patients who are PS0 or 1, and that the grade 3 toxicity was somewhat different, more nausea, more vomiting in the PS2 patients, but that it was tolerable. And of course, these patients desperately need a response if they're going to have any quality of life or length of life. And here you can see relative response rates. The PS2 patients did respond well to FOLFOX, and if you look at infusional combination therapy versus bolus therapy, they did better with infusional therapy. So they have a poor prognosis, but they seem to benefit from better therapy, and infusional therapy did give an advantage for them. Now, the other thing that happened in this study, somewhat by accident, is one day I got an email from Howard McLeod while he was working in Aberdeen. And Howard McLeod is one of the individuals who has pioneered pharmacogenetics. He was trolling around for phase 3 trials in which to get samples of germline DNA from patients. And I came across the 9741 trial and said, you know, this would be a really good trial for us to look at, because there are 2 arms of the study that have oxali in it, 2 arms of the study that have irinotecan in it, and 2 arms of the study that have 5-FU in it. So that's really an ideal study to look at the pharmacogenetics of the population with respect to 5-FU, irinotecan, and oxaliplatin. And we were able to incorporate this such that we got about 500 of the 1,700 patients who gave germline DNA to us to study. And as a consequence of this, we found that polymorphisms could predict response and toxicity, outcomes before treatment, an interesting study by Jeff Sloan, and who would get severe oxaliplatin toxicity. We also did an interesting study looking at Caucasian versus African American patients enrolled in the study, and I'll give you some info about that. So we looked at a number of different genes and their polymorphisms that were relevant to the metabolism of irinotecan, 5-FU, and oxaliplatin. These are genes relevant to irinotecan metabolism. You'll recognize UGT1A1 because that's the one that hit pay dirt in this study and others. Here you can see that the role of UGT1A1 is to glucuronidate SN38. SN38 is the active metabolite of irinotecan. It has to be glucuronidated to be excreted in the bile. As you know, there is a polymorphism, the 6,6, 6,7, and 7,7 polymorphisms in the American population, with about 10% of patients having an inefficient metabolization of glucuronidation process for irinotecan. And those patients have Gilbert's syndrome, something that was previously just of laboratory interest but not of biological interest. And they are at risk for irinotecan toxicity. So here you can see that those with the 7-7 polymorphism had about a 40% likelihood of getting grade 4 or 5 neutropenia as compared to the patients with the 6-6 and the 6-7 polymorphisms. And this actually went into changing the irinotecan package insert. Here you can see data looking at the other 4 genes, and note that ABCB1 correlated to response, the CYP genes to toxicity, and UGT also correlated with toxicity. Now, this led many individuals to look at UGT1A1 and irinotecan dosing. And what you can see here in this analysis that we did, there were some irinotecan regimens that looked at a very high every 3-week dose of 350 milligrams per meter squared, as opposed to the lower dose regimens more commonly used, the dose in FOLFIRI being 180 per meter squared every other week. And you could see that high doses correlated with toxicity, particularly for the 7-7 patients, with 83% of patients getting toxicity who were 7-7 getting the 350 per meter squared. So we sort of came up with this algorithm that if you are going to use the 350 per meter squared dosing, you have about a 1/3 chance of severe toxicity. You may want to go ahead and order this test, which you can order from a number of laboratories around the world. While if you're going to use lower dose irinotecan, the odds are much lower, and you may want to just treat and see how the patient does. Now, what about oxaliplatin genes? You'll recognize ERCC2 and XRCC that have been talked about in earlier talks today. These are genes related to the metabolism. Here you can see oxaliplatin being managed by these excision repair genes and detoxified by the GSTM and GSTP genes. And here you can see that levels of ERCC3 and the polymorphisms correlated with response, while GSTP correlated with incidence of neuropathy. Here you can see the response rate for the patients who did not have the GG polymorphism was higher. Interestingly enough, the GG polymorphism patients did better with irinotecan-containing regimens. Here you can see data on GSTP and grade 2-3 neuropathy, showing again that neuropathy was more common with the TT polymorphism. So this could potentially be useful in choosing treatments, where, for instance, people who had this polymorphism would get oxaliplatin. People with the CC polymorphism of GSTP would get irinotecan because they had a high risk of neuropathy. People with these 2 polymorphisms were both at risk for neuropathy and other toxicity, and then the majority of patients this technique would not be useful for. Now, this was a fascinating study that was done by Jeff Sloan and was presented at the plenary session of ASCO, looking at preliminary evidence of a relationship between genetic markers and oncology patient quality of life before treatment. So the hypothesis was that there are people that have higher quality of life based on genetics than others, and who have better resilience with disease and with chemotherapy for disease, and that we could identify them. So, we looked at the same genetic markers, 22 genes that we had picked mainly because those genes were relevant to drug metabolism. but some of them were also relevant to folate metabolism, to DNA synthesis, and to neurotransmitter metabolism. And you could essentially characterize them here as having different roles. So the bottom line was that we could make a case for effects of a number of these genes that we had looked at on general fitness. And when we ran this analysis, we found that we could correlate polymorphisms in a number of these genes with how patients were doing before they ever got chemotherapy. Now, unfortunately, no one has pursued this area of inquiry since that I'm aware of, but I think it's a provocative area that could be pursued. So winding up here, I'm going to talk a little bit about a study that we did on race in this. And it's been known that in colorectal cancer, African Americans tend to have lower response rates to conventional chemotherapy, and Asians tend to have more toxicity. We also know that there are gender differences, that women have more toxicity with 5-FU in terms of mucositis and neutropenia. So this was a study published in JCO in 2009 where we compared African Americans to Caucasians in terms of activity markers, chemotherapy toxicities, and the prevalence of polymorphisms. We allowed anyone who described themselves as African American to be in this category. About 10% of patients were African American across the various treatment regimens. You can see here that in all cases, if you pool all the data, African Americans had about a 30% response rate compared to Caucasians who had a 40% response rate. And this was biggest with FOLFOX, although FOLFOX was still the best regimen for both African Americans and Caucasians. And they actually also had lower toxicity rates, lower all-grade toxicity, and lower grade 4 diarrhea than did Caucasian patients. And as we looked at the polymorphisms in their genes, in virtually every gene we looked at, the profile of polymorphisms were different in African Americans than in Caucasians. And I'm just going to use UGT1A1 as one illustration. So while 47% of Caucasians had the 6-6 polymorphism, only 17% of African Americans did. 21% of African Americans had the 7-7 polymorphism compared to 9% of Caucasians. And if you look here at ERCC2 in another example, you can again see these great differentials, 70-30 essentially for the BB polymorphism of ERCC2. And so what we concluded from this study was that African Americans had a lower response rate. The differences in TTP and overall survival were not quite as consistent, but it was consistent that African Americans had less severe toxicity and different polymorphisms. So one could argue that in part of our individualizing of therapy, making race and the genetics that race is a surrogate for relevant may be important. So what did 9741 do in the 15 years since this study was conceived? Well, one of the things it did was to establish a new standard of care for metastatic colorectal cancer in the United States and lead to the licensing and approval of this agent, and hopefully led to helping many more patients than would have been helped with older regimens. I had an interesting conversation with Rich Shilsky recently where he said, it's gotten to the point where when a patient comes in with metastatic colorectal cancer and I give them FOLFOX plus this or that and they don't respond, that I'm outraged because we're so used to seeing responses or disease control. When I first started in the early 1980s, when all we had was 5-FU, if somebody's lung metastases responded to 5-FU, we'd leave the X-ray up for months because it was so unusual for somebody to get a response. This study helped to outline and underscore the importance of liver resection and the value of complete responses in metastatic colorectal cancer to the U.S. audience. It showed the benefit of treatment even in patients who had stable disease. It showed that elderly patients and PS2 patients can benefit from aggressive therapy. And it set this milestone of a 10% 5-year survival for patients with metastatic colorectal cancer, which certainly is a change from the days when I began my oncology fellowship. The study, because in part of Dan Sargent's scrutiny of it with respect to how it could influence clinical trials methodology, has helped us with the notion of real-time toxicity monitoring, has helped us to reduce the amount of adverse event reporting that we're required to do for regulatory purposes, has liberalized RESIST, and has perhaps pointed out the value of risk-stratified analysis. It's also helped to explain patient heterogeneity. If there's been one theme of this meeting, it's been patient heterogeneity. While Sabine is depressed by the notion that there is so much chaotic information out there, I'm exuberant about it. Because when I started out in this, we thought all colon cancer was the same and it was all untreatable. The fact that we now understand that there are so many different variations and that we can begin to impose some order on this chaos that will lead us to make smarter choices and better choices for our patients over time is really exhilarating. I would say that we've focused most of our research in heterogeneity on the tumor, and I still am hopeful that pharmacogenetics, which focuses heterogeneity on the patient's DNA, will also become more important to us over time with respect to quality of life, with respect to making smart choices about drugs, and for helping to understand the differences in population genetics across races and countries. And I think what this has pointed out is that the more people that you invite in to the circle, the more you get out of a trial. This was a large randomized cooperative group trial. We invited the quality of life people to the table. Howard McLeod invited himself to the table, but I was glad he did. And we invited many different statisticians, translational scientists, and clinical oncologists to analyze the data and use the data for various purposes. And as a consequence of that, I would say that those patients who enrolled in this trial leveraged their enrollment. They may have personally gained from it, but they also contributed to the science of clinical trials and to the science of managing patients with colorectal cancer by signing that consent form and sticking with the study. And so the obvious point about this is that the pathway to reduce the impact of cancer is through randomized clinical trials. One of my favorite philosophers is Yogi Berra, who said, if you come to a fork in the road, take it. Clearly, having a new diagnosis of metastatic colorectal cancer is a branch point in anyone's life, and our ability to enroll them on clinical trials, particularly smart clinical trials, is the way we're going to continue to make progress. Thank you very much for your attention.
Thank you. It's a remarkable collection of studies from one clinical trial. It's quite amazing. We have time for a couple of questions, if anybody's. Yes, Mark.
Just a very significant trial. Are the pathologic specimens available for molecular analysis?
So sadly not. When we embarked on this trial, we had lots of discussion about that. At that time, FNA was the way most people with metastatic disease were diagnosed. We didn't have the molecular techniques that we have now, and so we didn't make an effort to have tissue. Now, in the 15 years since then, we would never launch a trial like this again without getting that tissue, but that's part of the learning that has occurred in that time.
Your data in relation to genetic polymorphism and quality of life are quite provocative. Do you feel that the reason why there hasn't been follow-up in relation to that and in other studies is partially true to how you would design such a study? Maybe also the fact that N9741, because it's a study that has had such development over the year, that it's a mature trial, or is it really trying to decide what genes we should look at?
Well, I don't really know the answer to that. I have to say that it appears that pharmacogenetics, the interest in it peaked quickly. For a while it was the hot topic everywhere. And now it's harder to find trials that really inculcate it into the fabric of the trial. And I don't know whether that was because of the disappointment where something like the UGT1A1 seemed to be of more academic than practical interest. I think it's premature to abandon it. I think in part it's cooled because the funding agencies, which initially were very high on it, said, well, we're not getting a big return on our investment. We're going to focus on funding genetics that's oriented toward the tumor rather than toward the patient.
Rich, in saluting you on the McClay lecture and the superb one you've just treated us to, I thank Patty and all those who went into the choice of you to receive this great honor. The thing that also attracted me was that you were able, within the trial, to get Pharmacia to agree to someone else sitting at the table called Sanofi, and Sanofi Aventis, I guess Sanofi at the time. And that in itself must be a whole story that maybe is best told over Guinness. But this is our challenge, is it not? Is that when the funding is there that comes from industry very often is reluctant to, or has been in the past, to open a seat. Any insights or you can provide for all of us, including those in the audience who are from industry?
Well, what I would say about this is that, the leaders from Pharmacia were troubled, to say it mildly, when oxaliplatin found its way into this trial, and suddenly they were in a head-to-head comparison. Having said that, they funded the trial, and they continued to fund the irinotecan for the study throughout the course of the trial. It did lead to big switches in the market in terms of sales of colorectal cancer drugs. And I think to some degree that's why industry is cautious about these sorts of interactions. On the flip side of it, you could say that, well, it led to great advances in the management of colorectal cancer, and that's good for society, and therefore ultimately good for the companies. So I think it's a balancing act, and there are many, many complexities in this. I think to some degree Sanofi has continued to be very successful, and Pharmacia is now a part of Pfizer. And that was a consequence to some degree of this, and that's a reality. There was an incredible amount of politics about this, and I don't think I'll ever tell that story, even after a few Guinness.
Okay, I'd like to draw this session to a close and thank Professor Goldberg again for...
Thank you for the honor.